So our next topic is the digestion of nucleic acid. Okay, so nucleic acid, as you guys all know, present in the cell. We are eating many food, and where you can say that the nucleic acids are present. Okay, so nucleic acid will be in the form of DNA or RNA. Okay, if DNA is there, we will be saying that this is a deoxyribonucleic acid. Okay, if there is RNA, we will be saying that this is a ribonucleic acid. What does it mean? What's the difference between them? The difference between them is just of this one is having a 5 oxygen and this one is having the 4 oxygen, just that. Okay, one oxygen is less in the DNA, that's why it is called deoxyribonucleic acid. Okay, if you're talking about digestion, so in buccal cavity or in mouth, you can say in buccal cavity, new nucleic acid will digest because of no enzyme. As you guys all know, we were having no enzyme in the buccal cavity. So in buccal cavity, the digestion of nucleic acid will not occur. After moving through in the form of the bolus, it moved into the esophagus. After the peristalsis movement, it will move into the stomach. In stomach, we were having a no enzyme of the nucleic acid. Okay, so in stomach there will be no digestion of the nucleic acid. After moving through the intestine, which is the first part of the small intestine, that is duodenum. In duodenum, we were having a bile as well as nucleases, which was in the pancreatic juice. But if you're talking about the bile, so bile was having no enzyme. If you're talking about the pancreatic juice, which is in the involvement of the duodenum, so they were having a pancreatic juice. We were having an enzyme that was nucleases. As you guys all know, I have discussed that I will be discussing in the next video. I said to you guys in the previous video when I was discussing the detailed lecture of the pancreas. So in this, nucleases were defined in the two categories. One was the ribonucleases, which will work on the RNA, as well as this is a deoxyribonucleases, which was working on the DNA. Okay, so in RNA and DNA, nucleotides are there. In nucleotide, as you guys all know, there is a ribose sugar plus nitrogenous base plus phosphate group. Okay, if you're talking about the nucleotide will further break down into the nucleoside. I'm just giving you the concept. Okay, this is in which the ribose sugar is present as well as only nitrogenous base. Here the phosphate group is absent. Okay, so for example, if you're talking about the DNA, on DNA the enzyme will work, that is deoxyribonucleases. Okay, in the previous, here we have written that deoxyribonucleases will work. Here the ribonucleases will work. So here the deoxyribonucleases will work and it will break into the deoxyribonucleotide. As you guys all know, I said you guys that DNA will first break down the nucleotide in the presence of the enzyme. So it is further break down into the nucleotide. If you're talking about the RNA, so in RNA, the same enzyme will work, ribonucleases, and it will further break down into the ribonucleotide. Okay, now if you're talking about the pancreas, this is present in the pancreatic juice. Okay, this enzyme. Now if you're talking about the intestinal enzyme, number one enzyme we are having nucleotidase, and second one we are having a nucleosidase. Okay, Now further nucleotide will break down into the nucleoside in the presence of this enzyme which is in the intestinal. Let's see here, this is form which is deoxyribonucleotide. Nucleotide will further break down by the nucleotidase, DNA nucleotidase. Okay, we are talking about this DNA, and it will further break down into the nucleoside. Okay, if you're talking about the RNA ribonucleotide, here this is nucleotide. It will further break down by the nucleotidase. But what RNA? It will also break into the nucleoside. Now on nucleoside this enzyme will work. Which enzyme? Nucleosidase will work further on this enzyme. Now if you're talking about the nucleosidases, so further this nucleoside will break, nucleoside of DNA, nucleoside of RNA will further break by the nucleosidase. Let's see how. Nucleoside of DNA, nucleoside of RNA, it will further break by the nucleosidase. Okay, this will be same, but here DNA will be there and DNA nucleosidase will be there, and here the RNA nucleosidase will be there. The nucleoside will be further break down in the nitrogenous bases plus pentose sugar. Okay, in nitrogenous bases there are purine and pyrimidine. In at DNA there will be the adenine, guanine, cytosine and thymine. Here this will also break into the nitrogenous base plus pentose sugar. Okay, as well as here also the purine and pyrimidine are there, but if you're talking about the bases, which is here the adenine, guanine, cytosine, and as you guys all know, in RNA the thymine is present, and in behalf of thymine we are having the uracil, so it will get easily digest in the duodenum. The digestion will complete in the duodenum. So here we have discussed about the digestion of nucleic acid. Here you can see that how the enzyme have break their they into the small parts. Okay, so today we have discussed about the digestion of the nucleic acid.